Hi all, and welcome to this webcast entitled Human Embryonic Stem Cells to Treat Retinal Degeneration. My name is Sandra Petrus-Reurer, and I'm a postdoctoral researcher working at the Department of Clinical Neuroscience at Karolinska Institute under the supervision of Professor Anders Cavanta and Associate Professor Fredrik Lanner. Today, I will talk about my doctoral and postdoctoral research that aims to pave the path to the clinics using cellular therapy for patients suffering from a specific type of retinal blindness. In fact, here on the left, you can see an image that shows the vision of a healthy individual, the vision that most likely most of you watching today has. Whereas on the right, you have an image of the vision of a person that suffers from age-related macular degeneration, or AMD, which clearly represents an impaired central vision. This disease in particular, and these patients, of course, are the main focus of our research. If we go back to the organ and the tissue where the disease roots from, we have to travel to the eye and the retina. So if we go in a little tour, the light goes through the pupil and it gets scattered by the lens to the back of the eye, where it finds the retina. The retina is a complex tissue consisting of several layers of cells, going from neurons and photoreceptors, which are in charge of capturing this light stimuli and transmitting it to the brain, to retinal pigmented epithelial cells, or RPEs, which will be the main character of this talk. And these are cells of pigmented nature, as you can see in this histological picture on the right. RPEs have very important support functions for the photoreceptor cells, including its involvement in light absorption. They also are important for nutrient uptake. They have a very important role in the visual cycle. They also secrete angiogenic and immunosuppressive factors such as VEGF and PDF, and they have a very important functions of phagocytosis, of recycling. By phagocytosis, these outer segments that the photoreceptors produce, and that in case of not being recycled, these segments will be super toxic for the whole retinal tissue. RPE cells sit on a basal matrix called Bruch's membrane, and all this together is embraced by a vascular bed called choroid that gives the oxygen and the nutrients that are needed for these cells and for this tissue to remain functional. So what happens in case of disease or age-related macular degeneration is that because of not really well-described reasons, the supportive RPE layer, specifically in the macular region, starts degenerating. And as a consequence, the photoreceptors that deeply depend on these RPE cells also start to die off, then generating this vision disability in patients that start seeing a black spot in the middle of their field of view, which increases at the same time and speed as the degeneration progresses. So more advanced stages of this dry form of the disease are known as geographic atrophy. And as the name indicates, the degeneration is well demarcated by this gray area that you can see in this fundus image. Geographic atrophy accounts today for more than 50 million cases in the world, and the worst is that currently there is no really treatment available. So the approach that our lab took is to try to repair what is damaged or lost using human embryonic stem cells as source of RPE cells. Pluripotent human embryonic stem cells derive from the inner cell mass of an in vitro fertilized egg that is at the blastocyst stage, and these cells have the potential to give rise to all tissues of our bodies except of the placenta. The inner cell mass can then be isolated and kept in culture as a cell line of human embryonic pluripotent stem cells that, with the right cues, can then be differentiated to retinal pigment epithelium that then would be transplanted into these patients. So human embryonic stem cells provide us with an unlimited source of genomically and telomerically healthy and young cells. Now, bringing a stem cell therapy to the clinics is not a trivial procedure, since it requires to go through several checkpoints and, most importantly, to get green light in all of them. Checkpoints that include 1, having a xeno-free defined and functional protocol, 2, that is also safe and pure, 3, that is robust and scalable, and 4, that is immunocompatible for successful transplantation, and also 5, that is able to be converted into GMP clinical grade to finally have the derived RPE cells ready for testing in a clinical trial with patients with AMD. So I will now go briefly through the efforts that our lab made in order to overcome each one of these checkpoints and get an almost ready out-of-the-shelf RPE product. So we first developed a protocol that relied on embryonic bodies that were escaped from confluent human embryonic stem cells. These embryonic bodies, after approximately 8 18 days in culture, they start generating these pigmented optic vesicles, where RPEs would emerge from. These optic vesicles were then manually dissected and dissociated into single cells, and then they were operated on specific substrates to finally get stem cell-derived RPE cells. So the beauty of this protocol is that it's xeno-free and defined, which is a crucial step for clinical translation. In fact, it was the manual dissection, in addition to the use of the appropriate substrate, that was key to guide the cells towards this RPE lineage. And as an example, I want to show you an experiment in which we tried several substrates present in the native Bruch's membrane, ranging from gelatin and also several laminins from BioLamina, including laminin 111, laminin 332, laminin 511, and laminin 521. So we could see that 30 days after dissociation, it was laminin 521, the one that gave the best support for homogeneous expansion of these pigmented RPE cells. So we decided to move forward with laminin 521. 60 days after the first scraping, we got cells that really resembled the native RPEs with a cobblestone and tie morphology, but also pigmented, as you can appreciate in this bright field picture on the left. These cells also show the expression of RPE-specific markers, such as bestrophin-1, cis-retinal aldate binding protein, also known as CRALP, and also RPE-related markers, indicative of their polarization, like the sodium-potassium ATPase, or indicative of their tight junctions, such as ZO1. We then also evaluated that these cells are functional in vitro. And among other tests, we assessed their phagocytic function that I mentioned before by incubating them with Pit-C-label photoreceptor outer segments. And that showed that at 37 degrees, the derived RPE cells that here are labeled with phalloidin in red were able to engulf these green outer segments compared to cells that were at 4 degrees, where all cellular processes are stopped, including any type of phagocytic activity. So we then managed to get a xeno-free and defined protocol that allowed us to derive in vitro functional RPE cells from human embryonic stem cells. Next step was a translation into a relevant preclinical model to assess survival, integration, and functionality of the derived cells in vivo. So for that, our lab decided to focus their efforts in a bit of a different model, a large eye in vivo model, or the rabbit eye. The reasons being the large size of the rabbit eye relative to its body, but also the similarity to the human eye that allowed us to use the exact same surgical and imaging techniques that would be used in a human setting down the line. So we then carried out subretinal injections of RPEs into albino rabbits with the advantage that the RPEs of these animals are transparent compared to the ones of human origin that, as you saw, they were nicely pigmented. These gave us a very easy readout to assess cell integration into an albino rabbit retina, as you can see in the picture below with the black patch of a 3 months transplanted human stem cell-derived RPEs. So upon histology at 2 months after injection, these cells also appear pigmented compared to the control retina and also show the expression of human NUMA marker, here seen as pink nuclei, and polarized expression of bestrophin-1 shown in green. Also, and very excitingly, 8 months transplanted cells were able to phagocyte photoreceptor outer segments, as is shown by this green rhodopsin staining, and rhodopsin being an essential molecule present in these segments. Then, another interesting angle was to see if the cells were able to rescue retinal degeneration phenotype. So previous studies in our group showed that the injection of PBS only created a degeneration of the retinal layers, as you can see here in these OCTs, and especially in this histological section of a normal eye compared to a PBS-treated one. This seemed to be specific to the structure of the rabbit retina, and we took it to our advantage, since we could then assess the level of degeneration when the derived cells were injected. To our surprise and excitement, 2 months after the injection of the derived RPE cells, we could see that the outer retinal layer thickness was around 80% the thickness of the control retina in eyes that the cells were integrated. Then meaning that we could rescue and induce retinal degeneration by cells that got successfully integrated. So this adds another green light to our roadmap now regarding cell functionality of the cells in vivo. Next was to move on the assessment of the safety and cell purity of the derived RPE cells. And although I will not get into much details of these studies, overall they showed that the derived cells were karyotypically normal, genomically stable, they did not form any teratomas in immunocompromised mice, they did not migrate to other organs when injected subcutaneously in mice or subretinally in the rabbit eye, and also they did not have any transcriptomic or proteomic reminiscence of a pluripotent stem cell. Altogether, it was very encouraging to see that the differentiation protocol did not introduce malignant instability and was highly pure. So in the quest of improving this EB protocol, we invested some extra efforts in developing a more robust and scalable 2D protocol, aided by the identification of an RPE versus stem cell specific cell surface marker, as the CD140B was. That was actually identified from a commercially available antibody screen of 242 cell surface candidates. So just as a validation example here on the left, and upon fact sorting of a heterogeneous culture of RPEs, followed by cytospinning and staining of both CD140B positive and negative populations, you can see that in the stainings and the corresponding quantification, that was the positive fraction, the one that was also staining positive for bestrophin 1 and also with the most pigmentation, therefore representing this RPE population. As you can imagine, the CD140B cell surface marker will then become key for GMP monitoring and quality control evaluation of the differentiated cells. Additionally, we developed a 2D monolayer protocol that mainly relied on the addition of activin A at day 6 after stem cell seeding on laminin 521 or laminin 111, and a replating step at day 30 on laminin 521 for an additional 30 days. The cells derived from this protocol show typical pigmentation and bestrophin 1 and CD140B co-expression, as you can see in these immunofluorescence pictures, as well as the characteristic morphology, villi, and polarized pigmentation shown by scanning and transmission electronic microscopy. Upon transplantation, these cells also integrated in the subretinal space of albino rabbits and stained positive for anti-human NUMA and the RPE marker bestrophin 1. The main advantage of this protocol is its differentiation robustness, which was tested for 7 lines, 3 of human embryonic origin and 4 of iPS origin, and also its scalability, given the fact that from 1 million stem cells as a starting point, one can treat up to 13,000 patients with a dose injection of 50,000 cells per eye. So this adds another green light regarding robustness and scalability for this now optimized xeno-free and defined RPE protocol. So at this point of the road, we have cells that are derived in a xeno-free, defined, functional, safe, pure, robust, and scalable way. But it's of major importance to mention that these cells will only last days after transplantation if the immunological makeup is not compatible with the one of the patient that receives them. And because of this, cell will then be totally rejected, not feeling welcome in the recipient's body, and cleared in no time. For this reason, we thought that it was very important to invest some other extra efforts on making them as invisible as possible for the recipient's immune cells. And with that in mind, we decided to design the following HLA escaping strategy to make immune-compatible human embryonic stem cells. Then, if cells are left as they are as wild-type cells, the derived RPE cells will most likely be recognized by both the innate and the adaptive immune system, call it T cells and NK cells, and then they will be cleared up. Simplistically, the way it works is that a specific T cell receptor will recognize either HLA class 1 or HLA class 2 molecules from the injected cells. CD8 positive T cells will then recognize HLA class 1 complex, and specifically CD4 positive T cells will recognize HLA class 2, and then rejection will happen if this HLA molecule is present and mismatched. On the other hand, the native immune system works a little bit different, and so for the NK cells to not kill or to not clear up, they need to recognize this HLA class 1 complex, because it serves as a flag to tell that that cell is from the own body. Then, if that complex is missing or mismatched, the recognized cells will be targeted for cleared up. So as you can see, it's definitely not an easy task to trick our immune system, although it's also true that it seems that it's to a big extent HLA dependent. So we decided to edit the stem cells that will be further differentiated then after into RPE cells by knocking out HLA molecules using CRISPR-Cas9. So first we targeted beta-2M, which is a key molecule that aids presenting this HLA class 1 to the cell surface. And then we targeted C2A, which is a master transcriptional regulator for HLA class II genes. Like this, HLA molecules will not be seen by T cells, and therefore the injected cells will be expected not to be attacked by them. Although the problem might still remain with these NK cells that will lack the recognition of this HLA class I molecule in the knockout cells, and then they might still get activated. Firstly, and very briefly, we derived the single knockout beta-2M RPE cells lacking the beta-2M molecule, as you can see here in the immunofluorescence staining compared to wild-type cells. You can also see that HLA class I remained present intracellularly, as beta-2M is only key for the surface expression of HLA class I, as I mentioned before, but its absence in the surface was actually shown by flow cytometry when compared to wild-type cells. Similarly, we also derived double knockout RPE cells lacking HLA class I and II molecules, and although HLA class I was still present intracellularly, as shown before with single knockouts, it was not present in the cell surface. Also, HLA class II was not detected when assessed by both immunofluorescence and flow cytometry compared to wild-type cells. Remarkably, it must also be said that differentiated RPEs from both single knockout beta-2M and double knockout cells did not show any irregularity in the differentiation or survival, and also showed characteristic morphology and pigmentation of the native RPE cells. So we then embarked ourselves in understanding the reactions of the immune cells, both T cells and NK cells, when they get in contact or in co-culture in vitro with the derived and edited RPE cells. So for T cells, we measured the amount of interferon gamma that was released by the cultures as a measure of T cell proliferation and therefore activation. In these plots, you can see in the x-axis that T cells from two different donors were stimulated with IL-2 and anti-CD28, and also mixed with or without RPE cells, and in different RPE T cell ratios. We could then see that when RPEs were co-cultured with CD8 positive cells, only wild-type cells, represented by this black bar in this plot, that don't lack HLA class I, make T cells active and proliferative at both 1:20 and 1:50 ratios. And when co-cultured with CD4 positive T cells, was only the double knockout cells that you can see represented by this gray bar now that managed to dampen down this activation because of the lack of HLA class II that CD4 positive T cells now specifically recognize. So for NK cell responses, we both assessed NK cell activation or degranulation by CD107 expression by flow and NK cell cytotoxicity by chromium release upon RPE cell killing. To our surprise, and as you can also appreciate in these graphs, the NK cell behavior was mostly driven by the donor rather than by the cell type, then being independent, it seemed, of the lack of HLA class I or both HLA class I and II. This unexpected effect might be due to the nature of the RPE cells and their known immunosuppressive character, maybe expressing specific NK inhibitory ligands that then would compensate for the lack of this cognate HLA class I. Lastly, when the edited cells were transplanted in the xenograft albino rabbit model, subretinally and without suppression, we could see, judging by the infiltration present in these OCT images, that all transplanted eyes with wild-type cells were rejected, whereas the eyes transplanted with either single knockout beta-2M or double knockout cells had a significant reduction of the rejection rate. In conclusion, the edited double knockout RPE-derived cells evaded both CD4 and CD8 positive T cell responses, did not increase NK cell cytotoxicity, and when transplanted in the large eye model without suppression, they reduced the acute inflammation, and although I did not show it here, they also delayed the appearance of anti-human antibodies present in the rabbit serums that received the edited cells. Therefore, adding another green light to our roadmap for making the derived cells immune compatible. So with all this in mind, and as a summary, patients with geographic atrophy suffer from a degenerative disease causing their vision impairment and with no available treatment at the present moment. The use of cellular therapy is today one of the most promising approaches to control both the progression of the disease, but also to preserve the retinal layers that are still healthy. So our group then contributed to this road to the clinics by generating a source of cells that is xeno-free, defined, functional, but also safe, pure, robust, scalable, and immunocompatible. And also recently, we derived a GMP clinical grade pluripotent stem cell line that will be differentiated to RPEs and used for future clinical trial in the coming years, with the hope of getting the final green light of our roadmap to help patients with age-related macular degeneration. With that, I would like to thank all the people involved in this work, starting from Karolinska Institute colleagues led by Associate Professor Fredrik Lanner, who were key for the in vitro characterization of the cells. Santeric Eye Hospital colleagues led by Professor Anders Kevanta that carried out rapid subretinal injections and OCT imaging, and collaborators led by Associate Professor Helen Kaipa, Dr. Evren Alitsi, Dr. Anna Falk, and Dr. Emily Nandroth for all the help with reagents and the immunological characterization of the RPE cells. I would like to thank you all for listening, and please don't hesitate to contact me via my LinkedIn profile or my email in case of any questions. And also, I would like to use this space to wish you all the best with your future experiments. Thank you.